Hello students. In previous session we have completed simple permanent tissues like parenchyma, collenchyma and sclerenchyma. In this session let us learn in brief about conducting tissues known as vascular tissues helping in water conduction and food conduction. It represents xylem and phloem. As they are heterogeneous in nature, they are composed of conducting elements, surrounded by nourishing tissue and mechanically supported by dead cells. Let us begin with the xylem. It is commonly known as wood. Xylem is composed of three elements. Xylem proper or tracheary elements. These tracheary elements are the dead cells. They are of two types, tracheids and trachea. Trachea are commonly known as vessels. Tracheids are smaller in diameter, short in length. They have pits on the lateral walls and perforations are very small. These are unicellular. They are found in lower plants like pteridophytes, even in most of the gymnosperms. But trachea are like the pipes. They have big diameter, elongated, multicellular in arrangement and they have big perforations. So conducting capacity is more. They don't have pit fields, means lateral openings are absent. Tracheids and trachea. These two are main conducting elements of xylem. The lumen is a cavity. It is empty as it will help in conduction of sap from root to leaves. The wall of the xylem vessels and the tracheids are lignified. They are very thick. Lignin deposition is specific. It is in the form of circles known as annular. It is in the form of coilings known as spiral. It is in the form of steps, look like compartments known as scalariform, or it is in the form of network known as reticulate. And there are some where lignification is everywhere, leaving small areas. They are called pits. And some pits are bordered, so pitted and bordered pitted. So I told you about the lignification in xylem vessels: annular, spiral, scalariform, reticulate, simple pitted and bordered pitted. Now these tracheary elements are surrounded by xylem parenchyma. It is the only living tissue found in the xylem providing nourishment to the tracheary elements, and xylem parenchyma is mechanically supported by xylem fibers. As fibers are sclerenchymatous in nature, even these cells are dead. These are elongated, known as xylary fibers. So total 3 components we have seen. Tracheary elements includes tracheids and trachea. Trachea found in higher gymnosperms and all angiosperms. Tracheids are found in lower plants like pteridophytes and most of the gymnosperms. Surrounded by living tissue, xylem parenchyma, mechanically supported by xylem fibers. Xylem developing in the early phase of the plant, known as primary xylem. They have narrow lumen. Xylem developing in the perennial plants like dicot. It is derived from vascular cambium, known as secondary xylem. Primary xylem, which develops in early phase of the life of the plant, includes 2 types, protoxylem and metaxylem. Protoxylem early developed with narrow lumen. Metaxylem later developed with wide lumen. Depending upon the arrangement of proto and metaxylem in the central axis of the plant, we call xylem is endarch or xylem is exarch. Endarch xylem, protoxylem is towards the pith, metaxylem is towards pericycle. This condition we found in the stem, while exarch xylem is opposite. Metaxylem is towards pith and protoxylem is towards pericycle. This is found in the roots. Secondary xylem, a derivative of vascular cambium, which are developing seasonally, called as spring wood and autumn wood. Together, it forms annual ring. So every year, one annual ring develops. So by counting annual rings in dicot plants, we can find out the age of the plant. This is about the xylem, which will help in conduction of sap from root to shoot. The next complex permanent tissue is phloem. Phloem is commonly known as bast. This phloem composed of three elements. Sieve elements like sieve cells and sieve tubes. Sieve tubes are always associated with the companion cells. Sieve cells are found in lower plants, while sieve tubes are associated with companion cells found in the higher plants. Both are living components of phloem. They have thick cell walls, but in the lumen, it is not empty, it is carrying the solutes from leaf to different parts of the plant body. These sieve elements are surrounded by phloem parenchyma, and phloem parenchyma is mechanically supported by phloem fibers. Now in these three elements, only fibers are dead, while in xylem, only parenchyma is living. Here, if you see the sieve tubes at the ends, it is provided with the sieve plates with minute sieve pores through which we can see the protoplasm moving from one cell to the other cell. Like xylem, early developed phloem we call primary phloem. This primary phloem is known as protophlem and metaphloem, while the phloem developing during secondary growth of the dicot plant is called as secondary phloem. It is derivative of vascular cambium. Xylem and phloem representing vascular bundles. If xylem and phloem are separated from each other, arranged in the form of rings, it is the arrangement found in the roots of monocots and dicots. This arrangement is known as radial vascular bundles. If the xylem bundles and phloem bundles are countable, means they are minimum in number, either 4 or 6, it is said to be tetrarch or hexarch. And if number of xylem and phloem arranged on different radii are more, it is known as polyarch. Tetrarch or hexarch condition is the feature of dicot root, while polyarch condition we found in monocot root. When xylem and phloem are together, it is known as conjoint. It is on the same axis, so collateral. Conjoint, collateral, open, or conjoint, collateral, closed. Xylem phloem separated by a middle patch of dividing cells known as cambium. When cambium is present, it is said to be vascular, open vascular bundle, and if cambium is absent, it is said to be closed vascular bundle. So open bundles are found in dicot stem and closed bundles are found in monocot stem. So conjoint, collateral, closed, monocot stem. Collateral, open, dicot stem. There is one more type of vascular bundle we found in the family, the plants included in family like Apocynaceae. These bundles are known as bicollateral, where xylem is in the center of the bundle. It is on either side having cambium, so bundle is open, and on either side of the cambium we can see the phloem. So in other words, I can say xylem is sandwiched between the phloem and the cambium. These bundles are known as bicollateral. There is another type of vascular bundle we call concentric, means one vascular tissue is in the center and other tissue is surrounding the central tissue. For example, xylem is in the center and surrounded by the phloem. This concentric vascular bundle we call it as hydrocentric, endoxylic, or amphivessel. There are different names. And reverse, if xylem surrounding the phloem, so central portion is of phloem surrounded by xylem, it is known as amphivessel or leptocentric. So I told you about radial vascular bundles, conjoint collateral open vascular bundle, conjoint collateral closed vascular bundle, bicollateral vascular bundles in concentric, amphivessel and amphicribal. These are different types of vascular bundles we can see in the central axis of the plant body. So in this session we have completed the plant tissues including simple permanent, complex permanent. In the next session, I will brief you about the tissue system. It is the arrangement of these tissues forming the root and the shoot and the leaf. With this, I'll conclude this session. Thank you very much.